Welcome to the Translational Addiction Podcast, where we decode the innovative research from the Lotviepor Lab at UC Irvine, dedicated to translational addiction studies and developing effective treatments. Today, we got to sit down with Dr. Emily Castro, the primary author of Neuroglia in Substance Use Disorders, which was published in the Handbook of Clinical Neurology. It was also the review that Tanvi and I discussed in our second podcast. Dr. Castro is a postdoctoral research fellow at UC Irvine's Department of Emergency Medicine, where she investigates the molecular and behavioral mechanisms underlying substance use disorders. She earned her PhD in pharmacological sciences from UCI, where her research focused on the role of the human chRNA6 3' UTR polymorphism in nicotine-related behaviors and neurodevelopment. Her work integrates cutting-edge techniques such as CRISPR-Cas9 genomic editing, ribosome sequencing, and behavioral assays in humanized rodent models. Dr. Castro has authored multiple peer-reviewed publications in nicotine and tobacco research, neuropharmacology, and pharmacological research, and has presented her findings at major conferences including the Society of Neuroscience. She is a recipient of the 2024 Chancellor's Award for Distinguished Mentoring and has been recognized with honors from the Ford Foundation's NSF and NIDA's ICANN training program. Beyond the lab, Dr. Castro is a passionate advocate for diversity in STEM, having served as an outreach coordinator for UCI's School of Pharmacy and led initiatives to support underrepresented students. Her commitment to mentorship, community engagement, and translational addiction research makes her a compelling voice in the field, and we are so excited to share her insights with you in this episode today.
Okay, well, I can introduce myself first. I'm Michaela Simorton. I'm a fourth-year neurobio major at UC Irvine, and I was really interested in this lab because I don't know, I just wanted to learn more about like the mechanisms behind addiction because I've spoken with a lot of people who have either suffered from addiction or know people who have struggled with it. And yeah, I just wanted to learn more about the neural mechanisms, I guess.
Yeah, and I am Thanvi. I go to school in Philly. I am a third year and I'm studying neuroscience. And I guess I got into addiction research and stuff because Philly's obviously hit really hard by the overdose crisis. And so that in conjunction with harm reduction volunteering I've done in the city and I've always thought natural sciences were really cool. So the neurochemistry of it all was always cool to me. So that got me kind of into working with the lab. And I'm from Irvine, so I thought Dr. Lovebefort's work was really topical. But would you mind also introducing yourself and telling us a little bit about your scientific journey and career so far?
Yeah. Wait, one quick question. What
Mhm.
university in Philly?
Oh, I go to Penn.
Oh, okay. My friend in my cohort went to Drexel, so I was just wondering.
Oh, wait,
Um,
that's right next door. Yeah.
yeah. Yeah, so I actually started as a transfer student. So I went to Irvine Valley College, which is pretty close down the street. I changed my major, I don't know how many times, but I transferred to UCI as a psychology major actually. And while I was doing research as an undergrad in more cognitive science type labs, looked at some did some human work in Alzheimer's and different memory works. And I just had a lot of questions where the grad students would start telling me and teaching me about wet lab work, which means like working with animals or on the molecular side. And they're like, your questions are very molecular rather than behavior or humans, really. Have you ever thought about a wet lab? And like I barely know what that means. And I was psych major. I was like, does that happen? Do people do that? And so I was just really lucky in terms of having grad students that really encouraged me, working with grad students that really encouraged me. Of course, you can do anything you want. You can ask any question you want to and just reach out. So I got my first research as an undergrad in a wet lab experience was actually in a neurodevelopment lab that looked at the development of the auditory system in mice. So that was my introduction to molecular work and rodent work and this side of neuroscience. And then I realized that I was really interested in the brain and how it works and how it relates to behavior. And so then I was accepted into the UCI Interdepartmental Neuroscience Program, it's called. So I was able to, even with my psych degree, was able to get into the program. And they offer, it's an umbrella program. So they offer lab rotations where you do get to test out different research areas, human or rodent work. And so then my interest at the core were just neuroscience, neurodevelopment, neurodegeneration. And so I was very open to whatever field that led me into. And my first year was 2019 to 2020. So I was choosing a lab the week lockdown happened. So
Oh my
that
gosh.
really halted, yeah, that really halted everyone in their tracks, obviously. And then on the research side, it was like human work was really halted. We're really on the fence, and faculty and everyone was worried about their research and what would that lead to and what was going on. So I and a couple other members in my cohort were in a really sticky spot of like reaching out to faculties, and things were shut down, and no, we can't even physically have you in lab, and I don't know the money situation. And so I was also very fortunate that Dr. Lafavore had just received a really big grant from the TRDRP, so the tobacco research related disease. So he just had a good amount of funding, and he is just a really um, kind and great soul. And I was like middle of lockdown, and I was like panicking for a lab spot, and I reached out, we talked, and he gave me a shot. Where I really need to emphasize, there were so many PIs that were like, we can't physically have students in here, and just how important his work was, he was able to say, oh yeah, we still have research going on, and yeah, of course, like come try. And um uh yeah, I was able to, that was my introduction to the addiction side of research also. So um that is how I joined the lab and started here. And so I did my PhD here. We work with a very translational rodent model where we actually have a human genetic variation that we use CRISPR CRISPR genomic editing to introduce into a rat line. And my research focused on evaluating how that gene might alter protein expression in reward-related regions. And um, I have since stayed on to be a postdoc, and so I've been here for about a year now and kind of continued on some of the work that I found there. Um, and yeah, so I'm just really grateful to be able to say that, I think I really ended up where I was meant to be, and I've had a great time, have a great team. And so, yeah, so I'm happy to talk to you guys.
That's awesome. I feel like it's rare to hear people who transition between like behavioral and wet lab research, as well as like different topics. I feel like everyone's usually so hyper specialized, or like
Yeah.
from undergrad
Yeah.
onward, they like continue to specialize. So it's cool that you've had so much like flexibility in what you've explored. But I guess I also want to ask, like, you mentioned what it was like trying to get into a lab during lockdown. So I guess I was wondering what like being flexible and dealing with like unpredictable things in science looks like for you, especially in the context of like deep like funding cuts and stuff happening today.
Yeah.
Because I guess here like a lot of our labs are having funding cuts, and so they can take on way less students, which is obviously going to have like a downstream effect of how many people get to go into research and things like that. So I want to ask like what dealing with unpredictable things like looks like for you right now.
And that ended up being a foreshadowing, a preview for the rest of my time in the PhD program, to be honest. I had been told when I was searching for grad programs that, one of the biggest like signs and think qualities of a scientist that they look for at an early stage is adversity, diversity, and resilience. So
Hmm.
being adaptable, being able to like be so certain about your hypothesis and your experimental design and being able to listen to what the data says to adapt and pivot and change and integrate new information and move forward is science. So it's it's really hard, and some things are so out of your control. But it is really important to not um give up, really. And it's you hear that and you know that, and but in practice it's really hard. And so even that was really a taste of the rest of the time there of I have a plan and I work so hard and I thought of everything, and everything like things come out of left field, and you just have to move forward any way you can. Of course, funding is such a crucial fundamental. Like, how do you move on from no money? It's literally it's hard, but I do think that um, people are doing what they can to adjust their research questions, and adjust the focus to what we can, but also to just um, advocating for your science and advocating for the importance of it is also part of the job. So no matter what's going on or what is being the obstacles that are being placed, I think all the scientists are getting together to really try to advocate why certain topics and certain science can't be cut. So
So true.
that's
I
Yeah.
think that this podcast taught us that like the natural science component is as important as like communicating and advocacy and like showing people what your work means. I feel like
yeah,
both of them are equally important components. But yeah, thank you
Yeah.
for that.
Definitely. Well, I also wanted to ask, like, throughout your like maybe earlier, I guess, experimentation, did you ever feel kind of unsure or like have doubts in your hypothesis or your data was like kind of contradicting what you thought? And how did you deal with that?
every day, all the time, every
Yeah.
because like you
Oh my
spend
gosh.
so much time, especially when you're writing out the proposals for the work you're going to do, right? There's sections you're supposed to say these are potential pitfalls, expected outcomes, future direction. Like that's the title, but what you're really supposed to do is think about, although I have so much rationale and evidence and I'm expecting these results, what does it mean if I don't see what I expect to see? And it doesn't, the hard thing is keeping the mindset that that doesn't mean, you know, you trash your data. That doesn't mean you did it wrong. That doesn't mean I did something wrong. That doesn't mean you have to listen to your data. As long as you can trust yourself that you have your proper controls, you have thought through what your experimental groups should look like and what the question that you have and how your experimental setup reflects your question. If you've done the hard work and done the sitting and thinking through those aspects of it, you have to listen to your data. And so if it's not what you anticipated, what does it mean? And and it's hard because you're like, well, I already did that work. You have to do it again. And that's just the simple that's the simple fact. And I um I was like a young scientist and just student and just even my age too, just trying to not take it personally or trying not to I'm not my data. I am not my data. I just telling
Yeah.
myself that just negative data doesn't mean I'm a bad scientist. That type I will give all the props I can and just unwavering thank you and appreciation and gratitude to Dr. Lockwood in terms of fighting through or moving past that type of um, difficulty. He's very positive. He's a very positive person. He's really great at um, looking at your data and not just saying, oh, it's a no. He is really good at keeping perspective and saying, you know, it might not be what we expected. What does it mean? This is still cool. What else can it tell us? And another person that I have on just, I think you guys have met her, but just so much gratitude. And I'm so grateful that I've also been able to work with Dr. Frances Leslie. She was the prior dean of grad division. Her too, just bottomless fountain of wisdom. She is also been I don't know where I would be without her. But she's really good at that too. She's amazing and did a lot of mentoring and teaching me to let the science talk to me and let the data talk to me and follow the what the data is telling you, and keeping an open mind. And I do think that's really important because we get attached to our data,
Yeah. Oh my
even when you
gosh.
don't expect it. So,
Oh, I feel like I've noticed that even just in the lab meetings when people present, how their feedback is very constructive normally.
yeah.
It's not like tearing anybody down.
No,
Um,
I don't know, she's been a mentor for so long, and so
yeah.
she taught me to on not taking things personally, and I feel like it's really easy when she really doesn't give criticism or feedback in a personally, attacking kind of way. Some
Yeah.
science can be that way, or scientists can be that way. She also taught me the feedback sandwich. I don't know if you guys have heard of that, but
Yeah.
there's like a positive feedback, constructive and or negative, or this is what was negative about it.
Yeah.
That doesn't mean we're putting you in a negative light. This is just what can be improved, and then the positive feedback at the bottom too. So,
Yeah.
um, I see that in practice, and I practiced it with her as well. So, yeah,
Yeah.
great group,
Um,
great group.
That's so good. It's so interesting how much like the way you present feedback is impactful because like it could be the same thing, but it's just like the way it's presented to
Mhm.
you is more yeah. But oh, also we wanted to ask about um your work on neuroglia and how or like that paper that we just did our previous podcast on and what was it like kind of working on that and was there anything that stood out to you in that line of investigation?
Yeah, so that review was actually very special, and I'm glad you guys went over that. So it's special to me and special for, I think, a scientist at the level I was at. But I mentioned that my first wet lab experience was in an auditory focused lab in neurodevelopment, right? And so actually what I was able to do in that lab was learn about neuroglia as well. So that was the first introduction. And I really have to give context. I was told we were going to look at glia, and I'm saying that I was a psych major. I did not know that there are even these different cell types in the brain for me to even understand
Oh
them,
my
like have
gosh.
context. So that's the level I was at. And so glia became like near and dear to my heart in terms of science. And so then my work in as a PhD student didn't necessarily focus on glia, but the opportunity to work on this review paper came up, and they were interested in I can't remember the specifics of the call, but I do think they were interested in neuroglia. And so then it was actually Dr. Leslie who got invited to write this review, and she brought it up to me and Dr. Leffort. And she was like, Glia, I know you like Glia. Do you want to do this? And I was like, yeah.
Oh,
So special and near and dear to me. But really difficult at the stage I was at because this review was actually cleared to physicians. So I had to also learn a whole new perspective on how to convey that science because I'm still learning how to take my science and convey it to other people who do my work or who are in the same field or take my work and convey it to undergrads and like you guys, I want you students to know exactly what I'm talking about. So I'm trying to figure that out. And then this was a whole added layer of you're talking to physicians. They want to know the mechanisms of the medicines and what they impact and know the science behind that and
Hmm.
what that means for addiction patients or substance use disorders. So that was a special opportunity, I would really say. And so it was really difficult because then I was just collecting a lot of knowledge about medicines and how they have impacted receptors and what we see in the animal models and what we see in humans, and then speaking to physicians. So I had to learn that vocabulary and that how to communicate these things in a way that is helpful or useful for them. And so um, that was a really fun, um, difficult, challenging, but fun time just reading about that science. And I learned a lot. I feel like if it was enlightening to you guys, it's really because it was enlightening to me to understand that um, glia and mechanisms involved in glia can be treated and used as therapeutics. Um, it was really a deep dive that I went into and to understand those things.
Absolutely.
Yeah. Yeah.
It's such a like prolific review. It's like so instructive. I feel like when me and Michaela were reading it, just like the density of information, like I can see how that would be such a helpful resource for people looking
Yeah.
for treatment. Like there was a lot we had to like omit from the episode because there
Yeah.
was just such a like great density of things there.
Mhm.
Um, do you have a favorite like fact about glia you wanted to share? You said that you think a lot about them in terms of teaching and stuff. Is there anything interesting that stands out to you about them or that you like
Well,
about them?
I would really want to say something that's specific to the paper,
Go
to
ahead.
the paper that we talked, but my favorite fact, which I do talk about in the paper, but became it's such a foundational paper for the lab. It's not this one, but a different one. Where one of Dr. Leslie's students, her name is Dr. Kay Linker. She People understood that that microglia in adults didn't nicotine does not activate microglia in adults. That's what they understood. And in adults, nicotine is potentially anti-inflammatory, if anything. Um, and so while she was a grad student, she did an experiment where she measured um, microglia levels uh, in adolescent rats that were treated with nicotine. She found that they do induce a pro-inflammatory response. They do activate microglia. So that's a completely opposite effect between adolescence and adulthood. And then she also found behavioral parallels where that amount of nicotine, when given to adults, doesn't really increase or impact how much cocaine they take, but adolescents, given that same dose, it'll enhance their cocaine intake. So it's just a fundamental and such a specific impact and mechanism that she was able to identify, and just really highlights why we do a lot of our work in the lab is looking at the unique effects of drugs when it's adolescence versus adulthood, and that really the word I'm looking for, that really inform why nicotine can be so dangerous and other drugs can be just so uniquely dangerous when a brain is still developing, when a brain is still forming those crucial reward pathways, and why we do the work that we do, right? To really highlight this danger in adolescence versus adulthood. And I also mentioned, circling back to, I wasn't really sure what I wanted to look at, but I was very interested in neurodevelopment versus neurodegeneration, and truly being able to kind of look at both. They're two sides of the same coin. And so that's why a lot of this work is really interesting to me. And that's one of my favorite microglia tidbits.
Yeah, absolutely. That adolescent adult distinction is like what inspired me and Michaela to like dive into that paper for our
Yeah.
episode. Because we thought it was so fascinating within the like huge density.
Yeah.
Obviously, we were like, we definitely need to highlight that because it's such an interesting mechanism. Because I feel like we've heard so much about like the gateway drug effect, but we've never actually thought about what like in like what cellularly that means. So
Yeah.
I'm glad you pointed that out. And I guess we also wanted to ask you what is next for you scientifically, or like what's exciting or on your mind right now in terms of science, whatever you want to share.
Hmm. I could say in terms of science right now, in the position I'm in the lab, truly, it was like the hardest concept for me to wrap my head around, going from psych to molecular. It's the central dogma, and as you guys, especially like fully dedicated neuroscience majors, like I think you guys get a lot of training and you don't realize it, like just very strong foundations. So I say central dogma, and you're like, "Yeah, we do that all the time. And I really did it. That's like me joining this lab was my crash course in central dogma. I'm really wrapping my head around it. And I bring that up because this just the crucial idea of gene to DNA to protein or DNA RNA protein um is the foundation of a lot of the work that I'm doing right now. And it's a really genetics is just such a powerful tool that um we're still learning what to do with, you know? So a lot of what I'm doing right now is looking at translation activity of our nicotinic receptors, which is where nicotine binds to in our brains, and especially in adolescence, knowing the differences of how how that happens at the receptor level is really important for us to understand. And we have our humanized rodent model that will also inform us, where we already know that we have sex and genotype dependent differences in terms of nicotine exposure. So a lot of my work currently is looking at um, indirect measures of translation activity, because the nicotinic receptors are actually really difficult to parse apart. They liked to hang out as subunits all together. So really trying to delve into how the different subunits play a role is what I'm focusing on. And it's been really fun figuring that out. And just right now I'm at the stage of working on ribosomal sequencing. So since at the protein level, they're hard to really like parse away. We're looking at the ribosomal level, hoping that that indicates translation activity. Um, I'm getting that going in the lab, and hopefully that confirms some things that I was able to see in my PhD. And then, yeah, I'm still figuring out too what to do after that, after um, my time as a postdoc. It's a little bit of a tough time, but I'm excited. I'm just still learning about what the different opportunities are after here. Um,
Yeah.
yeah.
Oh, that's great. That's so cool. I really like your writing too. I think our next podcast is going to be nicotine on the developing brain. So it's another one of your papers. But yeah, I don't know, just like I enjoy your like I guess writing style is very clear and it's so dense, but it's like understandable. You know.
I took the role very seriously. Of She said, Dr. Leslie had said you want to walk your readers through your information and walk them through the message you're trying to convey. And it's
Yeah.
really hard. It's really hard. But I will say the
Yeah.
nicotine on developing brain was the first one. So I was
Okay.
definitely better the second time around with the neuroglia.
Okay,
Um,
okay.
but I do thank you so much. I do really enjoy writing too. That was actually one of the majors that I didn't follow through on as a at the community college level. But
Oh,
I really did enjoy English and writing, but I was just still figuring out what exactly was my calling or passion or what I saw myself doing long term. And it was just really just how life goes, that being a
yeah.
scientist involves a lot of writing.
It
So I
does.
was
Oh my
able to I
gosh.
was able to still do that
Yeah.
in my time here. Yeah.
Yeah. And like combine your interests. That's
Hmm.
cool.
Yeah.
Very good. Well, to close out a little bit, thank you so much again for your time. But do you have any advice at all for aspiring researchers?
I think really everything is an opportunity, even when it doesn't feel like it. So, I'm sure at your university as well, but I already know for UCI too, there's so many resources and there's so many events that happen. And I do think go every chance you can. Meet people, network, talk to other scientists. I think it's what you mentioned earlier too. Like there are a lot of scientists or just people that stay very narrow, or um, they focus on one thing, and it is your job to be an expert in a certain area. But I do think the power of science is when you start making connections and patterns and collaborations and really see the big picture on how methods in one field can be applied in your field and how you can use your expertise in a different area. I think those that type of mentality is a little bit harder to train or harder to instill or even not talked about sometimes. So some of my perspectives and ideas or just how systems work in the auditory lab that I was did help me navigate learning how systems communicate in an addiction model. So truly just everything's an opportunity and everything helps build your skills even when you don't see it. So keeping an
Yeah.
open mind and accepting different perspectives and those things are skills to practice. And yeah, and also being adaptable. Being
Yeah.
adaptable, being resilient, being um moving forward when you reach obstacles and challenges is a skill and it takes practice.
Yeah. It's such a like mindset sometimes I feel like if you're just putting effort into the things that you're
Yeah,
doing. Yeah.
yeah,
But that's cool that you've like gotten so far even if you wouldn't have like foreseen that
yeah,
it was gonna go in that direction. But
yeah.
yeah, I think that's all of the questions we had for you, unless you want to add anything else or any other like closing thoughts at all.
No, but not on my end. I kind of want to hear more about you guys. I just kind of
Yeah,
want
of
to know what
course.
you're interested in. You guys chose very neuroscience focused majors. What were you guys planning for after? And what are your long-term dreams?
Oh, totally. I really like or I am with the low beaver lab, but like here in Philly, I'm also with the child psych lab. So I do like the behavioral side of things. I'm actually not involved in like wet lab research, much like you weren't. Um, but because it like the rats and stuff kind of freak me out. But
Fair.
I like reading about it. I think it's
Yeah.
very cool. I just don't love being the girl behind the bench. But I work with like kids here, so like we do MRIs on kids. It's like a child psych lab here. So I see myself going to like medical school and potentially doing child psych or I don't know. I also have also like been really liking research recently, so somewhere between the two, I will have to figure out. But
Oh my goodness. When I was in the Alzheimer's lab and we did human MRI studies also,
mhm.
anytime I had a question about children or development, they were like, getting a child in an MRI is really hard. Like maybe don't have those questions.
Oh my gosh.
So
It's so
I'm happy
scary.
like things are like, I was like, but they're so valid questions. I was like, who's doing that? So
I
I'm like,
know.
yeah, it's happening and the
I
work is
know.
being done.
Oh my
It's so scary because the machine is so loud and they can't
Yeah.
like stay still for like an hour, which
Mhm.
understandable,
Yeah.
but absolutely.
Oh.
And what
Oh
about
my
you, Michaela?
gosh. Well, this is my first like research lab experience here, but I've done more of like EMT jobs or first responder, or I worked 911s for a little bit and just like as a lifeguard at the beach over summers. So um I don't know, I feel like I've worked with patients and I want to go to medical school. That's my
Oh,
goal. Um, but yeah, I just wanted to also get into the research side of like, I don't know, just having the opportunity to understand more in a more like guided and focused way instead of just me um trying to like unwrap stuff on my own. I don't know
yeah.
if that makes sense at all. But yeah, so I thought it was also really cool that the premise of this is to convey the scientific material to like even the general public or like just people outside of the field just so that they can like understand it and like through that it helps me understand a
Yeah.
lot more and it like makes it a lot more tangible than um oh like go read this entire research paper
Yeah.
um
Yeah.
yeah but that's kind of my
Yeah, something Michaela and I think about a lot is that like these papers like sometimes feel very like written for an audience by like that audience. Like it feels very insular, if, like only people
Yeah.
within research really are reading these things.
Yeah.
So it's really important to us, like whenever we work on this stuff, to not get super bogged down in stuff that like
Yeah.
somebody who's like passionate about addiction in like the normal world outside of science,
Yeah.
like would want to hear, you know?
It's something that I don't know if I've just had the right mentors or right, role models that's also been something that's really important to them. And so that was something that was brought up to me very early on of learning how to say things to someone who is not in science is really important. And in that line of thought too, I really hope you guys get a chance or set up talks with the grad students in the lab. And they're all really good in terms of we've all been trained that with that mentality too of like we want to convey why this is important to everyone and just science communication being so at the top of all of our interests. Yeah, I'll try to bug you guys on that too. I feel like they'd all be great to talk to. We all care very deeply about conveying our science. So please keep them in mind, all of them.
Yeah, I think that'd be amazing if we had maybe a little mini series of talking to the different lab members and just like
Yeah,
seeing their perspectives would be
yeah,
cool.
Totally, because
Yeah.
I feel like everyone has such a different journey in
yeah.
science and also like a different niche that they're interested in.
Hmm.
So
Yeah.
we that would be like so interesting to find out. Yeah. But yeah, thank you so much for like sitting down with us and talking to us and about your life and things like that. It was so interesting. And we also really liked reading your paper. So I'm glad we got like a little more insight into what inspired you to be a part of it.
Yeah.
Thank you guys. And you guys are
Yeah.
great. You guys, I know I already have the sense of you guys are gonna do really great things. And also just, yeah, I love this project of you that you guys have and just it's been great having you guys around. So thank you. And I please know that at the very least all of us are listening and it's not going into the abyss. We really enjoy these and this you guys are truly like we get the science out like this part of it is as critical, conveying the message. And you guys are going one step further and putting it in this format is really, really crucial for science
Oh,
as well. So thank you guys.
yeah.
You guys are a big part too.
Thank you. Well, yeah, we're excited to keep reading your work and seeing what you do.
Yeah.
So we will see you at lab meeting and things like that.
Yeah,
Thank you, Emily.
yeah.
Thank you
Thanks,
so
guys.
much.
Thank you all again for listening, and we hope this interview was as interesting as we thought it was. And thank you again, Emily, for joining us. If you're interested in learning more about the translational addiction studies of the lab, then please follow us on Instagram at Translational Addiction Pod for updates and check out the infographics and paper on the Lotfipour Lab website, which is linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipour Lab at UC Irvine, exploring how to understand addiction and develop effective treatments. See you next time. Thank you for listening.